This method can be used to assess how novel therapeutic agents or strategies influence or augment the cytotoxic T cell response during pancreatic cancer. This technique allows tumours to be rapidly digested and stimulated in vitro, allowing analysis of multiple parameters of the T cell response simultaneously, which can be easily adapted for other applications.
To begin, use forceps to transfer the tumor onto a petri dish. Store 5 milliliters of digestion medium in a 50 milliliter tube on ice to prevent enzyme activity commencing. Take a small aliquot of this solution to cover the tumor on the petri dish. Use a sterile scalpel and forceps to cut the tumor into small pieces, roughly less than 3 millimeters in length. Scrape the tumor pieces into the tube and gently invert the tube until all pieces are submerged in digestion media. Transfer the tube onto a shaking device for 20 minutes at 37 degrees Celsius to digest. Make sure all pieces of tumor are submerged and not stuck to the edge of the tube. Immediately after the digestion step, place the tube on ice to slow enzyme activity. Add EDTA to achieve a final concentration of 20 millimolar and briefly vortex the sample to mix in order to further slow enzyme activity. Open the tube and rinse any tumor digest off the lid of the tube with fresh RPMI medium. Then, prepare a 70-micron strainer, place it on a 50 milliliter open tube on ice. Pre-wet the filter with medium. Resuspend the digested cells and wash the sides.